Hi, we're the Smotherman Bat Lab here at Texas A&M University. Bats are the second most diverse group of mammals on the planet, and in our lab we study how bats use their amazing adaptations to sense the world around them.
Hi, my name is Kushal. I'm a graduate student in the lab, and I want to know if bat brains are doing anything different or special compared to brains of other mammals when they echolocate. Echolocation is where bats sense their world around them. They use sounds and listen for echoes of those sounds in order to get around. So here's the picture of a bat brain. And each of these little red dots is a brain cell called a neuron. And when I study the bat brain, I want to know how each of these neurons talks to each other to allow the bat to echolocate. So when I study echolocation, I use special speakers like these that can emit echoes to the bats, and we get to see how they react in response to the different sounds that we can play from the speaker. As bat biologists, we also use these special microphones that can detect the very high frequencies in the bat's echolocation calls.
Here's what echolocation calls look like, using special software that allows us to see and hear what these calls are. Let's take a listen.
My name's Allison, and I am a PhD student in the lab studying how fruit bats use their sense of smell to find food. As a field biologist, research to me means running around the jungle at night. Here is some footage of me collecting data in Panama in 2019.
Here's an example, day and night, of a bat biologist in the field. First, I would go into the forest during the day to look for good places to catch bats. Things I would look for would be open flyways and potentially fruiting trees that would attract fruit bats. We use mist nets to capture the bats as they fly through the forest. After untangling the bats, we put them into a soft cloth bag for transport. Sometimes we encounter animals other than bats out in the field, like this kinkajou frolicking through the tree branches. Once we've captured our bats, we bring them into the lab to begin our behavioral experiments.
Now what I'm working on is how bats use sense of smell to find food. So in order to do that, I am using an experimental setup in a flight cage in which I record the bats as they navigate to an odor source. So let's go ahead into the flight cage and I'll show you just kind of briefly what that looks like. So you'll have to excuse the terrible lighting, but these are the platforms which I have trained bats to pick a banana off of. So basically they're released into this room and I allow them to fly around until they find the platform that holds either the banana, or sometimes I use an object that is only banana scented. So right now you can see we just finished up a trial, so this platform is empty, while the rest of these platforms have little cosmetic sponges which serve as a control to account for their echolocation calls. So this is the video command center. So you can see that I have two cameras set up, one looking on that side and then one on that side. And this software lets me record the videos simultaneously, so everything is nice and synchronized, which I'll then use for creating 3D reconstructions of the flight paths from the bats. It's hard to see on the video of my video, but up here in the corner is where my bat is currently hanging out, and hopefully he will decide to eventually actually fly around. And naturally, the best part is these guys right here. So these are the Jamaican fruit-eating bats, Artebius gemmicensis.
At the end of the night and when the experiments are over, I give the bats a little bit of sugar water as a reward before releasing them in the area that we caught them.
So that's a summary of some of the work that we do in our lab. Here are some more fascinating facts about bats.
Bats belong to the order Chiroptera, which roughly translates as hand wing. That's because the wings of bats are basically elongated fingers of the hand with a skin membrane between the fingers. So that means that bats fly via the power of jazz hands. With over 1,400 species, bats are the second most diverse group of mammals, the most diverse group being rodents. All that diversity means that bats eat lots of different types of things and live in lots of different places. About 70% of the world's bats are insectivores, meaning they eat insects and other arthropods, like these Mexican free-tailed bats. Mexican free-tailed bats are among the most abundant bats in North America, particularly here in Texas. This is slow-motion video of the emergence from Bracken Cave, which is found in San Antonio, Texas. This emergence is sometimes affectionately called the batnado, for the way that the bats spiral as they emerge from the mouth of the cave. At its summertime peak, Bracken Cave is home to between 15 and 20 million Mexican free-tailed bats, making it the largest known colony of bats in the entire world. All these bats eat a lot of insects every night. It's estimated that the Mexican free-tailed bats in Texas save farmers millions of dollars every year in pest control services, particularly by eating the pests of crops such as cotton and corn. In addition to pest control, some bats also play important roles as pollinators. This is the endangered long-nosed bat, which is an pollinator for agave, which is a culturally and economically important plant in Mexico. Nectar-feeding bats use their especially long tongues to get at the nectar inside flowers. Meanwhile, other bats prefer to snack on fruit and are important seed dispersers by helping move seeds around the forest. And finally, the most famous of the bats, the vampire bats, which feed on blood. However, of the 1,400 species of bat, only 3 of them are vampire bats. Bats can live in a variety of habitats, the most famous being caves, such as these aptly named cave myotis. Bats will also roost in other places, like trees and even palm fronds, like this yellow bat. Other bats like to roost in man-made structures, like these bats at the Congress Avenue Bridge in Austin, or these bats spotted emerging from a temple in southern Thailand. We hope you've enjoyed your glimpse into the mysterious world of bats. You can learn more about our research by checking out our website and following us on Twitter.
Hamer Labs, we use a variety of tools to research the ecology of diseases that are spread by insects like mosquitoes and arachnids like ticks. These tools include molecular techniques, modeling, as well as field and laboratory studies. In this video, we will talk through 2 disease systems that we study and are here in the Bryan College Station area. One thing both these diseases have in common is they are transmitted between hosts, that is, people or other animals that can carry the parasite through a vector. Now, what is a vector? In this case, a vector is an insect or arachnid that is necessary to spread a disease between people or animals that can get the disease. In this picture, do you know which insects can be vectors? Now, from these vectors that can spread disease, have you seen any in your backyard or here in Texas? The vectors we are going to talk about today are the mosquito and the kissing bug, both of which can be found around here. West Nile virus is a flavivirus and is the most common mosquito-borne disease in the continental United States. People usually contract the virus from the bite of an infected mosquito. Luckily, only 1 in about 50 people develop serious, life-threatening illness after being bitten. Due to its prevalence, or how much of it there is in a population, it's important to understand its transmission and prevention. West Nile virus was first discovered in the United States in the year 2000 in birds in the Bronx Zoo. West Nile virus normally cycles between mosquitoes and birds. The virus can be passed to new hosts, including birds, horses, or humans, infecting them when the mosquito feeds. One way we study West Nile virus is by studying the birds and the mosquitoes. To study the birds, we have to catch, measure, and take samples from them. The most common way to catch most passerine bird species is using mist nets. As the name suggests, these nets are very thin and in the right conditions can be almost invisible to birds. Birds fly into nets, safely become entangled in its pockets, and are carefully extracted. We place a metal band on the bird's leg. We take a small amount of blood, less than 1% of the bird's body weight in grams, from the brachial vein in the wing. In this way, we can take biological samples from birds that can be used to test for a variety of diseases and conditions, including West Nile virus, and afterwards let the birds go. Nile virus in birds, so let's spend some time talking about mosquitoes and the types of research we do with them. To catch mosquitoes from the wild, we use mosquito traps. Gravid traps attract female mosquitoes that are ready to lay eggs. These mosquitoes are of particular interest when studying diseases because we know they have already fed on a host and have had the opportunity to become infected. BG traps use bait that smells like the human body to attract mosquitoes that feed preferentially on humans. Light traps attract the majority of mosquitoes and can be made more effective by placing a container with dry ice nearby. The dry ice releases carbon dioxide, which tricks the mosquitoes into thinking something is breathing nearby. When it gets close to the source of the gas, it's sucked into the trap by a fan. We can then use lab techniques such as PCR, gel electrophoresis, and stable isotope analysis to gather data about what host a given mosquito fed on, if it's infected with any pathogens, and how it moves in the environment. We can use this data to better understand how West Nile virus is spread in the landscape. We also use mosquito colonies maintained in the laboratory to research vector-host interactions and mosquito behavior. That wraps up our discussion on West Nile virus. Now, we move on to another vector-borne disease we study, Chagas disease. Chagas disease is caused by a parasite called Trypanosoma cruzi that is spread from animal to animal by kissing bugs. Chagas disease can affect the heart and GI tract of infected mammals, and around 8 million people have Chagas disease worldwide. Kissing bugs are present in Latin and South America and the southern U.S. Unlike mosquitoes, which pass West Nile virus to a new host through their saliva, kissing bugs spread the parasite through their poop. This means mammals can get the parasite when the poop gets into a cut or the bite site when the bug is eaten, which probably happens more by animals than humans, and through blood and organ donations, or to a fetus during pregnancy, which includes humans and our pets, dogs and cats. To study Chagas disease, we need to study the hosts, in this case the mammals that are infected with the parasite and the kissing bug vectors. To understand the mammal hosts, we have to go out and find them. Some animals are easier to find than others. For example, Chagas disease can be a problem for dogs. To study Chagas disease in dogs, we partner with dog owners to take blood samples from their dogs and study the parasite. Because Chagas disease can affect heart health, we also sometimes monitor a dog's heart functions to figure out what Chagas disease can do to dogs' hearts. To trap wild mammals, we go out in the evenings to set out mammal traps. We have small traps, called Sherman traps, that catch small mammals like mice and rats, and medium-sized traps called tomahawk traps to catch medium-sized mammals like possums and raccoons. If we are trapping when it's cold outside, like it has been lately, we make sure to put some extra padding in the traps so small animals can stay warm overnight. The next morning, we go back, find all the traps, and start working with the mammals. From each animal, we record what species they are and measure their sex and weight. We look for ectoparasites like fleas and ticks, just in case we can find any other vectors for other projects. We also sometimes will collect a small blood sample to test for the Chagas disease parasite. In the lab, we take the blood samples, extract out all the DNA, and test them for the parasite, Trypanosoma cruzi. If DNA from the parasite is found in the blood, or if antibodies to the parasite are present, then the animal is considered positive for the parasite. To study the vector, we go out and collect kissing bugs right when it's starting to get dark outside. Adult kissing bugs can fly and can sometimes be found on the sides of buildings, around porch lights, or in outdoor dog kennels. When we find a kissing bug, we only pick up the bug with gloves or a plastic bag so we don't directly touch them, since the parasite may be in the feces. Once we caught the kissing bug, we can either bring it back to our lab and test it for T. cruzi, or we can add it to our colony of kissing bugs on Texas A&M's campus. With this colony of over 1,000 kissing bugs, we test different hypotheses about the kissing bug behavior and their interactions with the parasite. We also have a community science program you can be involved in. On our website, you can find information on kissing bugs, like what adults look like versus nymphs, how many species there are, and where you can find kissing bugs in the United States. If you ever find a kissing bug, after carefully handling it, you can send it to us. First, send us an email with a picture of the bug you found. This is an important step because there are a lot of bugs that look similar to kissing bugs and are considered lookalikes that are also here in Bryan College Station. If it is a kissing bug, a member of our team will tell you how to send us the bug, and we can test it for T. cruzi, the Chagas disease parasite.
manager here at the Sea Life facility at Texas A&M University at Galveston. First off, my favorite sea animal is the cow-nosed ray. So I hope you all love stingrays. Want to learn more about stingrays? Give me an email. So, but I wanted to talk to you today about what I do and why I love what I do here at the Sea Life facility. So I manage the facility, which means I get to work with researchers, undergraduate students, grad students, and of course, many amazing sea organisms like sea turtles. We work with sharks, fish, invertebrates like crabs, fiddler crabs, and fireworms and jellyfish, and most importantly, I get to help undergraduate students learn how to take care of these animals so they can go on to careers, whether it's in research, zoos and aquarium, or rehabilitation organizations. I was a student here at A&M Galveston. I was a marine biology and marine fisheries major, so woo! And basically I came out of school really interested in fisheries ecology, so that's why I did my master's degree in, with, of course, cow-nosed rays. But really it was just a great opportunity after I finished my master's degree to come back to here at A&M Galveston and again get to work with our researchers. So at the facility, we are responsible for the daily husbandry and care of our animals, so things like preparing feeds, doing water quality tests, and of course taking care of the equipment, pumps, filters, different types of machines and things like that are responsible for maintaining that water quality. Because of course, when we deal with marine and aquatic organisms, they live in the water, so we have to take care of that water.
So this is our main tank room of the Sea Life facility. We have lots of different sized tanks available for our researchers to use. We have 20 juvenile alligators, and we're doing salinity tests where we're actually exposing these alligators to low salinity and looking at how it impacts their physiology. Specifically, we're looking at how the saltwater exposure impacts their stress hormone levels. These alligators were provided by a refuge in Louisiana. In this room, we have a couple different experiments. So right now you're looking at some oyster spat experiments. Again, we are looking at how environmental stressors impact these oyster spat. Specifically, we're looking at how temperature and salinity affects the rate of growth of these oysters. Here in our coral tank, we have a couple different species of corals, actually with a couple different research projects. One project's going to be looking at how toxins impact corals, and the other project's going to look at how heat impacts the corals. And then in this tank here, we have some upside down jellyfish. So these are a Florida species of jellyfish that are commonly found in mangroves. And right now we're just learning about how to take care of them. These are several different species of blennies. These are cryptic coral reef and oyster reef type fish. We've been working with these species for 5+ years. Primarily with these blennies, we are doing breeding work. We're actually breeding these fish in captivity and then working with the larval fish. These guys like to hide out, so we have plenty of rocks and other structures available for them to take shelter and refuge in. Here's our phytoplankton wall, where we're culturing different species of phytoplankton that we feed out to our zooplankton that are then fed to the larval fish. Here's a fireworm. These worms are called fireworms because of all of those white chaetae that you saw. We have sheep's head minnows, which are used by our mariculture class. These students get to actually breed these fish in captivity as part of their laboratory work during the semester?
As far as my career path, so like I mentioned, I was a marine biology, marine fisheries major, but I took a lot of classes here at the university, a lot of chemistry. So again, the fact that we are studying and, in our case, taking care of animals in a marine environment. Water chemistry. So getting a good firm foundation in chemistry is often very helpful for marine biologists. And then I did a lot of activities like working at Sea Camp as an instructor, so getting to do educational outreach. As scientists, part of our job is relaying that information that we learn about and research about to the public. And so getting the opportunity to talk to folks like y'all, or older adults, or younger kids, or everything in between is also a really important skill, and one that was real fun to do throughout my career and even now. And then other opportunities, any opportunity is great. So there are lots of skills that are needed as a marine biologist. So whether that's educational outreach or learning how to take care of a boat motor. I wish I would have had more experience with mechanics in shops class in high school. So any opportunity that you have as you prepare for college is a great opportunity. So don't limit yourselves with that. And then advice for future marine biologists, go out and explore. Whether or not you're located on the coast like we are here in Galveston, or you're in a landlocked state, go out and explore nature. You can do science at any spot in the world, and so just go out, enjoy nature, learn more about those habitats and ecosystems around you, and all of that will be great experience coming into a college degree and becoming a scientist.
Howdy, we are the Helms Lab. So for Darwin Day 2021, we thought it was fitting to talk about an idea that Charles Darwin himself introduced in his first book, The Origin of Species. In this case, he was talking about the incredible evolution between an orchid flower and the bee that pollinates it. The orchid is entirely dependent on this bee to spread its pollen, or to live on and continue evolving. The orchid and the bee is an incredible display of coevolution, or one species affecting another species' evolution over time and vice versa. Now, coevolution does not just take place for orchids and bees, but between many different plants, insects, and animal species. Coevolution can be a result of predators eating prey, like a lion eating a zebra, two species competing against each other, like two birds who look like the same seed, and two species that help each other, like a bird eating annoying bugs off of a cow. Plants and insects have co-evolved in a number of different ways. Some of these are good and helpful to the plant, like pollination, while others are harmful or do not benefit the plant, like herbivores. Insect herbivores have co-evolved with many different plants in many different ways. Some insects are so specialized that they can only feed on one kind of plant, like a squash, while others can snack a little bit on a lot of plants. These plants, when being fed on, have co-evolved some defenses that can protect or minimize the damage done by these insects. And insects have co-evolved ways to get around these plant defenses. This coevolution is a constant battle, with both attempting to win over the other. Now, coevolution leads to one of the most important topics about communication. Communication among plants and insects. You see, plants may not be able to talk with sounds like us. They use a different method, which is essentially they release volatile organic compounds, also known as VOCs, into the air, like talking through smells. And these VOCs don't just reach plants around them, they also can have the insects pick up on them. So these smells are also released above ground and below ground by plant roots. The smells that plants release can change depending on what's going on with them or their environment. So I want you to think for a second about the smell of grass after it's mowed. So this smell is essentially the grass's way of yelling, "Danger, danger, I was attacked," to the other grass around it. For a chemical or scent in the air to matter to an individual plant or insect, there needs to be some ability to recognize and associate that smell. Think of how some of your favorite smells stick to you and why. When cookies are baking in the oven and you start to smell the sugar in the air, you get hungry. In a similar way, plants and insects can associate certain smells with certain meanings. But how can plants smell when they don't have a nose? You see, they smell through pores all along their skin, microscopic openings called stomata. Insects don't have stomata, but they also have little pores on their antennae usually that allow them to smell. You see, there are smells that are particular for some insects. And so, in some plants, they've picked up how to attract parasitic wasps, which will attack the insect herbivores, and they attract them by releasing this particular smell. And so this is just one of the really cool ways that plants have co-evolved with insects to protect themselves against insect herbivores.
Hi, my name is Natalie and I work on corn. In this lab, we're interested in how plants understand their environment, and sometimes they do this by smelling things around them. So in my studies, I look at plants that look pretty healthy and have very open pores to smell things around them. And I also study plants that are more dry and will have closed pores or stomata, and how this might affect what they are smelling around them. Thanks.
Salt marsh moth uses the smell of the pumpkin to locate the plant.
So this little caterpillar, if you watch, it's just traveling slowly towards it, but when it gets closer, it gets a bit more confident, and it's trying to decide, what is the easiest and quickest way to get a tasty snack. And so the more VOCs in the air, the easier it is to target that plant.
Hi, I'm Laura Malejo, and I study entomology here at A&M. And what I'm recently studying right now is how plants actually defend themselves or protect themselves against their different herbivore pests, such as caterpillars or beetles. And so specifically what I'm looking for is to see if plants can actually get ready to protect themselves right as soon as they're eaten by bugs.
With Laura's research, she's trying to better understand what's going on with the plants when they're being fed on and what's going on with plants around them. So, like that little caterpillar, this little squash bug searches for the plant and it locates it. And it uses a particular smell that the pumpkin releases to identify it as a tasty food source.
Hi, my name is Morgan, and I'm a PhD student in the Helms Lab, and I study a bunch of different gourd species, such as these pumpkin plants here, but I also work with zucchini plants and watermelon plants and cucumber plants. And my research focuses on understanding how when beetle larvae or beetle babies feed on the roots of these plants, how they affect the scents or the smells of the leaves of the plant, and how then that changes how the plant is communicating with above ground insect herbivores. So I've done a lot of research with squash bugs, which are an insect that feed on the leaves and the stems of squash plants and other gourd species. And I've studied how below ground root feeding herbivores affect the above ground chemical cues and then influence squash bugs.
So those little beetle babies snack on the roots of plants, which damages the amount of water that the plant can drink up from the soil. And so it can be life-threatening if the plant doesn't take care of it.
Hi, my name is Emma Manuel, and I'm studying the interactions between plant roots and beetle larvae, which are beetle babies. So plants, when they are damaged by the babies, will emit a smell that attracts predators to come and attack the beetles. But what the beetles do is they actually can prevent the plant from releasing these smells and thereby avoiding being attacked. So I'm trying to understand these relationships between the plants and the beetles.
With our research in plants and insects, we study all different kinds of things. And so if you guys have any questions, we're going to be here after. And thank you so much for listening to this presentation.
Hello everyone, my name is Travis Richards. I'm a postdoctoral researcher here in the Wells Fisheries Lab, and I work in the Marine Biology Department. And I'm going to talk with you today a little bit about what I'm doing as a postdoc here at TAMUG. And so I have here today some example organisms that we collected from the Gulf of Mexico, the deep sea environment, over the course of 4 years. And I just wanted to geek out a little bit about the deep sea and tell you about some of the really cool animals that we actually have here in our backyard. All right, here's a little sample platter of some of the species we get in the deep sea. And I pulled these animals in particular because they represent some of the dominant families of species that we have in the deep pelagic Gulf of Mexico. So I'm going to start over here in the top left with these two fish here. These are two different species that belong to the same family, and that family is Myctophidae, or the lanternfishes. And so lanternfishes are a group that are distributed worldwide. They are ubiquitous throughout the world's oceans. They are numerically one of the dominant groups of deep sea fishes, and that's important because they serve as an important sort of top-down influence in the food web because they eat zooplankton constantly. So they swim up to the surface at night where zooplankton are plentiful. They leave the deep sea and swim up under the cover of darkness to avoid predation, and they feed heavily on zooplankton, and then they migrate back down to the deep ocean during the day to sort of hide from their predators. And while they're down there, they are often eaten by larger species. And by swimming up to the surface, feeding heavily, and then swimming back down, they're actually an important vector of energy from the surface of the ocean to the deep sea. So they actually connect species from the surface, the zooplankton, with species down in the deep sea who maybe don't migrate up. And the lanternfishes get their name because, like a lot of deep sea species, they can create their own light, right? They use bioluminescence. And what's interesting about them is they actually have these bioluminescent photophores along the sides of their body. That might be kind of hard for you to see on both species. And many of the species of myctophids have species-specific patterns of these photophores, and so scientists believe that they can use these to signal to their mates what species they are. Hey, I'm friendly, let's get together, let's mate. Because you got to think, if you're in the deep sea and it's dark and you're creating light, yes, you could be attracting prey, you could be attracting a mate, but you could also be alerting a predator to your whereabouts. So they have this species-specific sort of light signal that they send out. And then this species down here, which is called Diaphis, is actually really interesting because if you look on the front of the face, you can see this sort of silvery section on its nose. That is actually a light-producing photophore. And so scientists think they actually use that almost like a searchlight to look for a specific prey using their own bioluminescence. So a very important and interesting group, the myctophids. Over here we have what is called a hatchetfish. You might have seen these featured on BBC's Blue Planet. It's another midwater fish. They also migrate vertically up through the water column at night to feed. But they have this nice silvery coloration that actually reflects the light around them and helps them blend in. And if you can look very carefully along the belly, it's kind of hard to see, they also have photophores. What's interesting about these photophores is they actually use these photophores to match the color of downwelling light to help them camouflage. So if you're a predator in the deep sea looking for prey, the way you look for prey is you get underneath them and you look up, because you're going to look for the silhouette that the fish's body makes because of the sunlight filtering down through the water column. And if you want to get rid of that silhouette, you need to make your body the same color as that downwelling light. And so what's interesting about these hatchetfish is they actually have, it's hard to see, they have a photophore in their eye that they use as a reference point. So that photophore tries to match the color of the downwelling light. The eye sees that, and then that signals to the belly what color to sort of make the belly photophores. And that hides their profile from predators looking at them from below. So they have basically a way to eliminate their profile or their silhouette to predators looking from deeper waters beneath them. So hatchetfish, very interesting group. This species over here is pretty fascinating. This is a type of dragonfish. This is more of a predatory fish. These are smaller examples, but they get relatively large for deep pelagic fishes. They can get up to about 12 inches long. And they eat myctophids. They feed predominantly on myctophids, and you can again see they have sort of these light-producing photophores along their jaws or along their face. And like many deep-sea predatory fishes, you can see how gigantic their gape is. Right? If I pull that jaw back, you can see it can open almost 180 degrees. And so they're very good at swallowing prey, many times sort of their head size. And a lot of deep sea fishes have that ability. It allows them to sort of take advantage of any meal when it comes by. So this dragonfish is an interesting example of what a predator in the deep sea looks like. Again, very elongate, not great swimmers, mostly a sit and wait predator. Another sort of dragonfish-like species is this bristlemouth. Bristlemouths are one of the family is Gonostomatidae. They are perhaps the most abundant fishes on the planet. They're found in all the oceans of the world, and they are highly abundant. And this is an interesting fish because they have really nice examples of these photophores all along their bodies. And again, you can see these bristlemouths, which eat crustaceans like these shrimp, have pretty huge and wicked-looking mouths with lots of backwards recurved teeth to sort of hold on to their prey. So an important consumer in part because they are so abundant. Now we get to some of the crustaceans. Crustaceans, numerically speaking, often match the abundance of fishes in the deep sea. Interestingly, almost all of them are colored red. So these two migrate up through the water column at night to feed, but if you remember from sort of your intro to oceanography class, red light dissipates the fastest in water. So if you are red colored, there's no red light and you appear pitch black in the deep sea. And so they come in all shapes and sizes. These are two extremely abundant species. This is Notostomius in the deep sea, and we have a larger specimen here. But some deep sea shrimp, not this species but a similar species, they also use bioluminescence. They don't use it in the same way that the fishes do. They don't have photophores all the time. Some do, but this one species actually uses bioluminescence in a very interesting way. It's called the spewing shrimp, and when it's sort of startled by a predator, it will actually sort of vomit or throw up bioluminescent liquid. It will spray it out of its mouth, and that will either distract the predator, or some people say it will actually stick to the predator's face, and then that predator now has like this bioluminescent marker which could alert another predator to its presence, and that predator can come eat the other predator. How often that happens, we don't know, but regardless, they use the bioluminescent sort of vomit as a way to distract or deter predation in the deep sea. So just a few examples of some of the cool animals we have right here in the Gulf of Mexico, and there are many, many more to talk about.